Here we go again, another Paper 2 series. Nonetheless, this one is the 9700 October-November 2010 Paper 22. If you are new to this channel, you know what to do. Hit that subscribe button, like, comment, and finally watch and learn. Without wasting further more time, let's get into it. Question 1. Protein production involves a complex sequence of events and a number of cell structures. The first column in table 1.1 shows some of the events that occur in the production of a protein in a cell and its eventual release from the cell. As you can see, the table over here is given. They have given the events and the sequence of events as well as the cell location. Part 1. In table 1.1, write the sequence in which the events occur, using 1 as the first process in the sequence. What is this sequence about? Well, it is about the protein production. And if you recall the steps of protein production, first is transcription, which occurs in the nucleus, where mRNA molecule is synthesized. Followed by translation, where the mRNA molecule binds to the ribosome as well as the tRNA molecule brings a specific amino acid that follows the complementary base pairing of the mRNA strand. After the process of translation is done, then you get a polypeptide or a protein. Following that, protein modification would take place, and that is number 3. Since this protein which is made has to be released from the cell, then it is concerned with exocytosis. And in order to release the protein from the cell, it has to be in a form of a vesicle. Hence, the next event would be secretory vesicle formation, being step number 4. And finally, exocytosis, which is where the vesicle binds to the cell surface membrane and the vesicle containing the substance, that is a protein, is eventually released from the cell. Hence, the last step being exocytosis, that is number 5. Again, the first step is transcription, second is translation, third is protein modification, fourth is secretory vesicle formation, fifth is exocytosis. Part 2. From the list A to F below, choose one cell location for each event and write the letter in table 1.1. Each letter may be used once, more than once, or not at all. The letters given are A. Golgi apparatus, B. Lysosome, C. Nucleus, D. Rough endoplasmic reticulum, E. Smooth endoplasmic reticulum, F. Plasma cell surface membrane. Let's first look at the first event, that is transcription. Well, where does transcription occur? Transcription occurs in the nucleus. And what letter is the nucleus? Letter C. Therefore, C belongs to transcription. What about translation? Translation occurs in the ribosome. And where are ribosomes present? In the rough endoplasmic reticulum. Therefore, D belongs to translation. Then, if you go to step number 3, protein modification. Protein modification occurs in the Golgi body. Hence, being letter A, Golgi apparatus. It could also be D, rough endoplasmic reticulum. Nonetheless, the best possible answer is A, Golgi apparatus. And step number 4, secretory vesicle formation. Where does secretory vesicle formation occur? It occurs in the Golgi body too. That is the Golgi apparatus. Hence, letter A belongs to both protein modification as well as secretory vesicle formation. And the final step, exocytosis, belongs to letter F, which is the cell surface membrane, as the vesicle binds to the cell surface membrane and then exocytosis occurs. Part B. Describe the process of exocytosis. Exocytosis is an active process that requires ATP, and it is a process by which materials are removed from cells by the fusion of vesicles containing the substance with the membrane. Finally, the contents are secreted out of the cell. So, the 3 key points that would earn you 3 marks are: it is an active process that requires ATP, process by which materials are removed from cells by the fusion of vesicles with the membrane, and finally, the contents are secreted out of the cell. Another key point you could mention is that the vesicle moves towards the membrane. But the main 3 key points are: it is an active process, vesicles are involved, where the vesicles fuse with the cell surface membrane, and finally, the contents are secreted out of the cell. Part C. One example of protein modification is removal of the first amino acid, methionine, from a newly formed polypeptide chain to make a functioning protein. The DNA nucleotide sequence that specifies the amino acid methionine is TAC. State the mRNA nucleotide sequence that is complementary to the DNA sequence for methionine. In complementary base pairing, A binds with T or U being only in the RNA, because in RNA T is not present. Instead of T, it is base U which is present. And also C binds with G, that is cytosine binds with guanine. Since the amino acid methionine is TAC in the DNA nucleotide sequence, the complementary mRNA nucleotide sequence would be AUG. Since T binds with A binds with U since this is RNA, and C binds with G. Hope that is clear. Part 2. Suggest 2 other ways in which the polypeptide chain is modified to produce the functioning protein. 2 other ways are, you could talk about the addition of sugars to proteins to form glycoproteins, because when you add sugar units to proteins, you get glycoproteins. You could talk about the removal of some other amino acids. Instead of the amino acid given over here, such as methionine, you could talk about there could be some other removal of amino acids in the polypeptide chain when it is being modified. Then you could also mention addition of non-protein components or prosthetic groups. One such example is heme being added to globin to form hemoglobin. Other key points would be formation of tertiary structure or formation of named bonds such as ionic bonds, hydrogen bonds, disulfide bonds. So since this is a suggestion question, you could write down any of the given points. Question 2. Malaria is an infectious disease that is considered by the World Health Organization to be a disease of worldwide importance. Explain what is meant by the term infectious. Always when they ask this kind of question, what is meant by the term infectious, you have to mention 2 important things. Infectious is when it is caused by a pathogen and it is transmissible. That is, it can be passed on from one person to another. So the 2 key points to remember are caused by a pathogen and transmissible. What is the meaning of transmissible? It means that it can be passed from one person to another. That would earn you 2 marks straight away. Part B. Name one species of organism that causes malaria. Malaria is caused by any of the given species that are Plasmodium falciparum, Plasmodium ovale, Plasmodium vivax, and Plasmodium malariae. Another side note you should keep in mind is that the causative agent of malaria is a protoctist, but the name of the species is Plasmodium falciparum or any of the others. Part C. Explain the significance of the following statements in the control of malaria. The female Anopheles mosquito has been more closely studied with regard to malaria than the male Anopheles mosquito. Question over here asks, what is the significance of this statement in the control of malaria? So the significance is that only female mosquitoes take blood meals, or they only require blood for egg development. Male Anopheles mosquitoes do not require blood for egg development. And you could also mention that only female Anopheles mosquitoes act as the vector. Part 2. The infective stages of the malarial organism are present in anticoagulant produced by the mosquito. It is because this is what is passed when a mosquito sucks blood from a person. The anticoagulant prevents blood clots, hence it is easier for it to obtain its blood meal. And part 3. After circulating in the blood for a short time, the pathogen enters liver cells of the newly infected person and then enters red blood cells. What is the significance of this statement in the control of malaria? Well, if it enters the liver, the parasite is concealed or hidden from the immune system and drugs would be unable to reach them. Therefore, this is a key thing to help scientists or whoever makes drugs that target the specific parasite know that they should target the pathogen before it reaches the host cells. It also helps understand that the parasite reproduces inside liver or red blood cells. Again, as you can see over here towards the end, provides the idea that the pathogen is only present in blood plasma for a short time, and in order to control malaria, you should target the pathogen before it reaches these host cells. Therefore, that is why this statement is significant in the control of malaria. Part D. Discuss the factors that determine the distribution of malaria worldwide. Distribution of malaria is mainly in the tropical regions and are found in areas where both the pathogen and the vector, which is the Anopheles female mosquito, are present. You could also mention presence of vector in hot and humid areas, therefore leading to more incidences of malaria. Other factors would be some areas could have already dealt with and eradicated malaria, whereas more cases in countries with poor health facilities or low economically developed countries. Disease could further be spread by people who migrate from regions where malaria is present or is endemic. Then malaria could be present in areas where mosquitoes are already resistant to use of insecticides, therefore determining the distribution of malaria worldwide. So these are some factors that could affect where malaria is found often and where it is found least. So you could write 4 key points from the list given over here, and that would earn you 4 marks. Question 3a. Enzymes are globular proteins that catalyze metabolic reactions. Describe the features of globular proteins. Key thing you should know, or key thing you must know, is that all globular proteins have a spherical shape and all globular proteins are soluble in water. Another feature of globular proteins are they all have a tertiary structure with hydrophobic groups facing towards the center and hydrophilic groups on the outer surface. How do you remember whether the hydrophobic groups are on the outside or in the inside? Well, just remember that the proteins are soluble in water. In order to be soluble in water, obviously the hydrophilic groups should be present on the outer surface because it is the outer surface that comes in contact with the water. Hydrophilic stands for water loving, therefore should be present on the outer surface. Hydrophobic stands for water hating, and water hating groups, known as the hydrophobic groups, point towards the center, that is away from the watery surroundings, and that gives enzymes its globular shape. So since the question asks for the features of globular proteins, you could mention they have a spherical shape or ball shape. Then you could also talk about that proteins are soluble in water, and you could mention that they have a tertiary structure with hydrophobic groups facing the center and hydrophilic groups on the outside surface. Part B. Enzymes can be used to remove cell walls from plant and fungal cells. The cells are incubated in a solution that contains a mixture of enzymes. Suggest an explanation for the fact that a different mixture of enzymes is required to remove the walls of plant cells compared to the walls of fungal cells. Key thing you should know is that enzymes are specific in nature and they only act on one specific substrate. And the idea over here is that the composition of cell walls in both fungal cells and plant cells are different in nature. Cellulose is in plant cell walls, and in fungal cell walls it is made up of a substance known as chitin. Therefore, different mixture of enzymes are needed to remove the wall of plant cells, as the enzymes acting on plant cells won't be able to act on the walls of fungal cells because they are both made up of different substances. And the explanation is all enzymes are specific in nature and they only act on one specific substrate. Part 2. Explain why, when plant cells are incubated with enzymes to remove their cell walls, it is important to maintain an optimum pH. What is the meaning of optimum pH? Optimum pH is where the enzyme works best at that given pH. Therefore, a key point is enzymes work best at the optimum pH. Then either side of the optimum, that is either above or less than the optimum, the activity of the enzyme decreases. Change in pH affects the bonds holding the enzyme's tertiary structure, such as the ionic bonds. That is also a key point. Change in pH affects the ionic bonds, as well as it could also affect the hydrogen bonds holding the enzyme together. Then if the tertiary structure is affected due to the bonds being disrupted, the active site could change shape, because the tertiary structure determines the shape of the active site of an enzyme. Hence, if the tertiary structure changes shape, the active site changes shape. And if active site is altered, then the substrate is unable to bind with the active site and the reaction cannot proceed. Therefore, that is why it is important to maintain an optimum pH. Part C. A student carried out an investigation into osmosis using red blood cells. Red blood cells were placed in sodium chloride solutions at 5 different concentrations. For each concentration, a sample was added immediately to a microscope slide and the cells were viewed using a light microscope for a period of time. The observations recorded are shown in table 3.1. Explain in terms of water potential and osmosis the results that the student obtained. In this table given over here, the 2 columns are concentration of salt solution as well as the observation of red blood cells. And if you go to the table, for both 0.0% and 0.4%, water enters the cell. How do you know that? Because as indicated by the observation, there is an increase in size because the cell swells in 0.0% and in 0.4% the cell increases in size. That means that there is a higher water potential outside the cell than inside. And then distinguishing between 0.0% and 0.4%. Which one has a higher water potential? Well, higher water potential is in 0.0% solution than in 0.4% because more water enters the cell in 0.0%, leading to it bursting. Whereas in 0.4%, it doesn't mention the red blood cell bursting. It just mentions it increase in size. Where the cell bursts, that means that solution has a higher water potential. Therefore, 0.0% has a higher water potential than 0.4%, and that leads to water entering the cell and changing in size. Then there is no change in size as indicated at 0.9%. Why is that? Because the water potential outside the cell and inside the cell are equal. Hence, no net movement of water takes place. That is how you could mention in terms of water potential and osmosis the results that the student obtained. If you look at 1.5% and 3%, there is a decrease in size or the cells become smaller. Why is that? It is because the water potential in the solution is lower than the water potential in the cell. Therefore, water will leave the cell and follow down the water potential gradient setup. And how do you know which one has a lower water potential, whether it is 1.5% or 3%? Lower water potential is in 3% solution than 1.5%, because as you can see from the number itself, 3% means it is more concentrated. That means it has lower water potential. Hence, more water leaves the cell in 3% solution and therefore more smaller and shriveled in size. That is how you explain in terms of water potential and osmosis the results that the student obtained. The key part over here is the difference in water potential outside and inside the cell. And in terms of osmosis, you should say whether the water enters a cell or water leaves a cell and why. For the why part, you should mention the difference in water potential gradient setup. Part D. The student also carried out a similar investigation using plant cells with cell walls removed. These cells were suspended in a 12% mannitol solution so that the water potential inside and outside of the cells was equal. The key part of this question is that the investigation was carried out using plant cells with cell walls removed. The student removed a sample of these cells. The sample was placed into distilled water and was viewed using a light microscope. Describe what you would expect the student to observe and explain why this would not occur with normal plant cells. Distilled water has a higher water potential than 12% mannitol solution. Hence, water enters a cell, cell increases in size and then bursts. Why does the cell burst? Because the question says cell walls have been removed. Although it is of a plant cell, if the cell walls have been removed, then there is no cell wall present to resist the pressure or forces building up, and it will burst, similar to the case of animal cells, because animal cells also have no cell walls, and that is why when water enters the cells, it leads to the cell increasing in size and eventually bursting. Similar case will occur over here as well, because no cell walls are present to resist the forces. And for the part, explain why this would not occur with normal plant cells. It would not occur with normal plant cells is because normal plant cells have cell walls, whereas this experiment right here is of plant cells with no cell walls. Question 4. Figure 4.1 is an incomplete flowchart showing some of the events of the primary immune response that occur after a person has been given a vaccine. Part A. Choose the correct term from the list below to complete figure 4.1. Lymphocytes, antigens, mitosis, vaccine, T helper lymphocytes, plasma cells, macrophages. If you look at this given figure, which is incomplete, a person is being given a vaccine. And what does a vaccine contain? A vaccine consists of antigens. Therefore, let's go through the flowchart. Terms to choose from are given below, so you don't have to think of what terms to fill in. Dead pathogen in. The best suitable answer is vaccine, because the dead pathogen is present in a vaccine. Followed by phagocytosis by. What cells undergo phagocytosis? Well, it is nothing but the macrophages. Hence, the best possible answer out of the given options below is macrophages. Cell presenting. What on its surface? It should be cell presenting antigens on its surface. Because antigens are present on the surface of the cell that consists of the pathogen, then lymphocytes would recognize and bind to these antigens. Therefore, recognition and binding by specific lymphocytes. And what is the purpose of lymphocytes? Lymphocytes, they produce antibodies. Hence, activation, then cell division by mitosis. because lymphocytes, they undergo clonal expansion by mitosis, and clonal expansion is nothing but cell division, but rapidly. Therefore, activation then cell division by mitosis, followed by plasma cells produce and release antibodies, because it is only plasma cells that produce antibodies. And out of the given options over here, plasma cells suits best. Therefore, plasma cells produce and release antibodies. Also, helper T lymphocytes secrete cytokines, and that is the only option left. And actually, the only cell that secretes cytokines are the helper T lymphocytes. Therefore, the T helper lymphocytes should fill that blank. And there we have it, the complete flowchart. Dead pathogen and vaccine, phagocytosis by macrophages, cell presenting antigens on its surface, recognition and binding by specific lymphocytes, activation then cell division by mitosis, plasma cell produce and release antibodies, helper T lymphocytes secrete cytokines. Part B. Explain why the person is unlikely to become ill if they are infected by the same pathogen some months later. The best possible answer over here is that the person already has active immunity, because if a person has active immunity, then only the person will not be infected by the same pathogen again. Hence, if a person comes in contact with the pathogen again, memory cells will already be in place that will stimulate plasma cells to be produced and more memory cells, because a person who has active immunity has memory cells in place, and memory cells, they form part of the immunological memory. Hence, if they come in contact with the pathogen again, they stimulate plasma cells to be produced rapidly and more memory cells. What does that aid in? Well, more plasma cells means more antibody concentrations, because plasma cells, they produce antibodies. Faster secondary immune response will occur as higher concentration of antibodies produced in a short time period. Therefore, the pathogen is destroyed before the person falls ill. Part C. Some parents decide that their children should not take part in a vaccination schedule. Suggest how a country-wide vaccination schedule can give protection against infection to unvaccinated children. Over here, you could mention the herd immunity. Herd immunity is where most people are already vaccinated. What does that help in? Well, it helps in reducing the pool of infected people in the community. Other key points you could also mention are less chance of transmission, reduced exposure to pathogen, protects those unvaccinated as disease does not spread. The two main key points are herd immunity. Herd immunity is a must over here, because if herd immunity is in place, then most people in a certain community are already vaccinated. If most people are already vaccinated, that reduces the pool of infected people in the community and less chance of transmission to those who are not vaccinated. Question 5. State the term that applies to each of the descriptions A to E. A. Storage polysaccharide in animals made of chains of 1,4 linked alpha glucose with 1,6 linkages forming branches. In animals, the only branch storage polysaccharide is glycogen. Therefore, the best possible answer over here is glycogen, because they are referring to the storage polysaccharide in animals and it is branched. How do you know it is branched? If it has 1,4 linkages as well as 1,6 linkages. And yes, glycogen is a polymer of alpha glucose, and yes, it does have 1,4 as well as 1,6 linkages. Hence, the best possible answer is glycogen. B. A plant that has adaptations to enable it to live in areas where water is in short supply. If you recall the chapter that deals with adaptations of plants, the best possible answer over here is xerophytes, because xerophytes are the only plants that have adaptations to enable them to live in areas where water is in short supply. Such adaptations are sunken stomata as well as presence of trichomes or hairs. This reduces the water potential gradient between the outer atmosphere as well as the leaf, hence allowing it to live in areas where water is in short supply. Now parts C, D and E, although they are not in the current syllabus, I have given what the answer is. C is haploid because one set is haploid, two sets is diploid. For D, the name of the trophic level to which photosynthetic organisms belong, the answer is primary producer. And for E, a process carried out by bacteria that involves the conversion of atmospheric nitrogen into nitrogenous compounds that can be used directly by plants is nitrogen fixation. Question 6. Figure 6.1 is a section through lung tissue showing an alveolus and its blood supply. Name the type of epithelial cell shown by label lines A and B. If the question mentions it is an alveolus, then the type of epithelium in alveoli or alveolus are squamous epithelium, and the cell would obviously be squamous cell. Hence, the name of the type of epithelial cell is squamous cell. Part 2. Describe how the elastic fibers of the alveoli contribute to the healthy functioning of the lungs. Elastic fibers, they aid in the stretch and recoil of the alveolus. Stretching during inspiration or inhaling, recoil during expiration or when you are exhaling. Stretches to increase the surface area and recoils to force the air out or expel the air out. This helps to prevent the cell from bursting. If elastic fibers are present, it ensures the cell doesn't burst when you inhale and exhale, because it allows the stretch and recoil function of the alveoli. Therefore, for 2 marks, writing down that it aids in stretch and recoil will give you 1 mark. The other mark is it stretches to increase the surface area and recoils to force the air out. Part B. The actual diameter of the alveolus along the line X to Y is 220 micrometers. Calculate the magnification of figure 6.1. Show your working and give your answer to the nearest whole number. Over here we are supposed to find the magnification, and magnification is nothing but image size upon actual size. And if you measure the given line over here, that is from X to Y, that measurement would be 75 millimeters. And if you convert it to the correct unit, that is to the same unit of the given question, that is in micrometers, and you substitute it into the equation given, which is magnification is equal to image size upon actual size. The actual size is 220 micrometers and the image size is 75,000 micrometers. You would get the magnification as 341 because you have to round it to the nearest whole number. Part C. Outline 2 features of a gas exchange surface that are shown on figure 6.1. If you look carefully at this figure 6.1, you will see that there are short diffusion distances, as well as the alveolus is surrounded by blood capillaries. Hence I have given over here short diffusion distance provided by thin alveolar wall, that is one feature. The other feature is surrounded by blood capillaries, ensures easy exchange of substances. Those 2 features will land you 2 marks, and it should be features that are shown on the figure, and these 2 features are definitely shown. Part D. Figure 6.2 is a simplified diagram of the circulatory system of a human showing gas exchange in the lungs and in respirant tissue. The partial pressures of oxygen and carbon dioxide at 4 locations are also shown. This diagram right here shows you the alveolus and the body tissue, as well as the partial pressures of the blood vessels and in the corresponding areas of the alveolus and the body tissue. So look carefully into the values of the partial pressures of oxygen and carbon dioxide, and let's hop on to the next question. With reference to figure 6.2, explain how the differences in partial pressures of oxygen and partial pressures of carbon dioxide in the alveolus and in blood enable gas exchange in the lungs and respirant tissue. Well, obviously the gases move around by diffusion. And how does diffusion take place? Well, diffusion takes place down a concentration gradient from high concentrations to low concentrations, or in this case from higher partial pressures to lower partial pressures. In the alveolus, if you look at the pulmonary artery, the partial pressure of oxygen is less than the partial pressure of oxygen in the alveolus, whereas the partial pressure of carbon dioxide is higher than the partial pressure of carbon dioxide in the alveolus. Therefore, oxygen will diffuse into the pulmonary artery while carbon dioxide will diffuse into the alveolus because of the partial pressure difference created. Whereas in the body tissue, if you look at the capillary network at the arterial end, the partial pressure of carbon dioxide is less than the partial pressure of carbon dioxide in the venous end, whereas the partial pressure of oxygen is higher than the partial pressure of oxygen in the venous end. And if you go to the key points given over here, in the alveolus, in the pulmonary artery, the partial pressure of oxygen is lower than the alveolar air partial pressure, whereas the partial pressure of carbon dioxide is higher. Hence, oxygen diffuses into the blood vessel and carbon dioxide diffuses into the alveolus. They both diffuse in the opposite way. And if you look at the respiring tissue, in the capillary network, the partial pressure of oxygen is higher than in tissue. Hence, oxygen diffuses into the tissue from blood, whereas the partial pressure of carbon dioxide is higher in tissue than in the blood. Hence, it diffuses from the tissue into the blood. Over here, you just simply have to look at the values given and then decide which way will the gases diffuse. And how do you decide that? You have to look at the value which is higher. For example, if you look at the capillary network, the partial pressure of oxygen is higher than in the tissue. Therefore, oxygen will diffuse into the tissue. Whereas the carbon dioxide is higher in the tissue than in blood. Therefore, carbon dioxide will diffuse into the blood from the tissue. So, you simply have to follow the path from high concentration to low concentration. And then this question becomes nothing but easy. And that is the end of this paper. If you did find this video useful, please do leave a like, comment and subscribe if you haven't already. More videos will be coming soon, therefore stay tuned for more.